Hi everyone, hi quarantined friends. Welcome to the lecture on section 6 of chapter 5, which is all on cell signaling. Cell signaling is crucial to cells. Cells have to be able to communicate with each other. If they didn't, that would lead to their demise or death. They have to be able to get signals from their environment, signals from other cells, and it's the combination of signaling molecules or ligands, if you remember from the cell communication POGL or Mr. Anderson, and receptors that are in the plasma membrane or cell membrane, if you remember that being called, these are receptor proteins that bind to the ligand and receive messages, and it's all about how cells then take those messages and respond to them, which really dictates their ability to be able to survive. So in eukaryotic cells, the communication can come in different ways. It can be direct contact. You might remember Mr. Anderson's example with the Post-it note. This is considered to be local signaling, where cells that are in direct contact or nearby interact with one another. You can also have cases of cell signaling, of local signaling, or the process that uses local regulators. This is a situation where the cells are not in direct contact, and a messenger molecule called a local regulator will travel a short distance to another cell. Animal cells have local regulators called growth factors. Sometimes you see that hormones as well. They can stimulate cell growth, division. Some cells respond to these while others don't. Some cells have the receptors for them while others don't. So here's some examples of local signaling and long distance signaling. So here they're calling it paracrine signaling. I know we've seen that from the cell communication POGL. Remember, that is communication between cells that are nearby each other. So these cells, this one in the middle here, is secreting local regulators, which would just be like little signaling molecules, and they're going to diffuse out, and the cells around them will receive them if they have the right receptors for them and do something with that message, whatever it be. The next one is synaptic signaling. This is another type of paracrine signaling, if you were referring back to the POGL from the beginning of the week. But it's a nearby form of cell communication. These little messenger molecules going in between these two neurons here are neurotransmitters. They're little chemical messages that send a signal. So this would be something like dopamine, serotonin. You might have heard of those if you're taking AP Psych. And then down here you have endocrine signaling. So this is usually with hormones. Hormones are secreted by the cells of your endocrine system, and then they have to travel a long distance to get to wherever they're going to go. In the example of testosterone, which is a hormone, a sex hormone, that will have to travel and bind to cells that are involved or can only receive that message and then do with that testosterone whatever needs to be done. We're going to look at an example of that. In both local and long distancing cell signaling, only specific target cells can recognize the signaling molecule, and that's a key theme throughout all of this. So they took the nervous system out of the AP Biology curriculum for this year, but if you are in anatomy or AP Psych, you've learned about this stuff. You've learned how neurons, which are the cells in your brain and in your nervous system, they exhibit a special type of local signaling called that synaptic signaling. So that was this one in the picture here. This is synaptic signaling. Okay. This is a case where the secretion of a chemical messenger, like a neurotransmitter, I gave you some examples, dopamine and serotonin, they will go across a synapse or a gap between cells and trigger a response. And you would consider that to be more of a local distancing or kind of a nearby signaling. Right, and this is what synaptic signaling looks like. Both animals and plants use hormones for long distance signaling. Cells will release the hormone molecules into their circulatory system, and they will travel throughout the body where target cells can recognize and respond to them. We do this with our hormones, and again, we're going to look at an example with testosterone in a little bit here. Plants also do it. They have hormones as well. For example, ethylene. Ethylene is a plant hormone that is released, and it causes plants to ripen. We're going to look at plant hormones and how plants communicate with each other probably towards the end of next week or the following week with more cell communication stuff. So, sorry, this is more on long distance signaling. We've seen that picture already. So with cell signaling, there are 3 basic stages, and these are 3 things that you would have to be able to know and explain, identify: reception, transduction, and the response. These are the three sort of stages that cell messaging is divided into. Reception is the chemical signal being detected by a receptor outside or inside of the cell. Transduction is the conversion of that signal to a specific cellular response, usually along a phosphorylation cascade or pathway. We're going to look at that. The response is whatever the cell has to do with the message, the actual thing or the actual response. All right, so I'm going to show you an animation of what that looks like.
Cells communicate with one another by means of chemical signals. For the receiving cell, there are three stages in the signaling process: reception, transduction, and cell response. The cell targeted by a particular signal has a receptor molecule complementary to the signal molecule or ligand. The ligand fits like a key in a lock and triggers a change in the receptor molecule. Signal transduction converts the change in the receptor to a form that can bring about a cellular response. This might involve a series of steps, a signal transduction pathway that alters and amplifies the change. In the third stage of cell signaling, the transduction process brings about a cellular response. This can be any of many different cellular activities, such as activation of a certain enzyme, rearrangement of the cytoskeleton, or activation of specific genes.
All right. I don't know why that guy sounds like he's doing the narration from inside of Fuzzy TV, but good animation, good explanation of this picture here. These are the three stages, or an overview of cell communication: the reception stage, the transduction, and the response. And we're going to go through all three in detail and kind of look at how they all work and what's going on here. All right, so starting with reception. The reception stage is based on the target cells that can detect and react to the hormone or the signaling molecule, often called a ligand. That's like the generic term for it, because certain cells have certain receptors either in the cell membrane, like in this picture here, or they're inside of the cell. We'll look at a picture of that shortly here. And once that ligand binds to that receptor, it's all hinging on its lock and key, right? The shape of that ligand and whether or not it can fit the receptor. And usually that will cause a conformational change or a shape change to this protein, which sends the message to the next part of the cell communication process. If you remember with enzymes, remember when the substrate binded to the enzyme in its active site, it caused that kind of conformational change, or they call it the enzyme substrate complex, kind of like a clasping handshake, right? Similar idea here. The protein's going to bind and sort of change shape, and that will send the cascade of signaling to the next part of the pathway. So signaling molecules can be lipophilic or lipophobic. And what that basically means is they can either cross the cell membrane or they can't cross the cell membrane. In the cell communication POGIL, I left number 11 as an extension question for you guys to answer. And it asked you why nonpolar molecules don't need a receptor protein to send a message. It's because they can go right through that cell membrane. And there are signaling molecules or ligands that are able to go right through the cell membrane, and they bind to receptors that are then inside of the cell. So there are two types of, we'll deal with those later, but there are two types of cell membrane receptors. These are ones that are right embedded in the cell membrane, and they function a little bit differently. So the first one is a G-coupled receptor. Mr. Anderson talks about this in the signal transduction pathway video that you watched. This is a case, or this is one of the types. The G protein coupled receptors work with the help of a protein that binds to an energy molecule that is very similar to ATP. It's called GTP. That's why they call it that. So here in this picture, you can see a G coupled receptor protein in action. The signaling molecule binds to the extracellular side of the receptor, that being this purple molecule here embedded in the cell membrane or plasma membrane. Okay, the cytoplasmic side of the cell, so that's this area in here, binds to and activates this G protein, which is carrying the GTP. So once this binds and activates, this now becomes activated, and the G protein leaves the receptor and it actually diffuses along the membrane and then binds to an enzyme, altering the enzyme's shape, and that activity then will activate that enzyme and trigger the next step, which is the signal transduction pathway. So this is one type of receptor in the cell membrane, a G protein coupled receptor. Another kind is a ligand-gated ion channel. If you've done neurons with action potential this year, you've seen this. If you haven't, that's okay. This is where you have a ligand-gated channel where regions of the cell membrane act like, actually they look like gates for ions. If you've done action potential in neurons, you remember the salty banana with the sodium on the outside and the potassium on the inside. Nerve cells function this way. They function with ligand-gated channels. You have to have receptors that assume a certain shape, and when that ligand binds to the gate, it opens or closes it, which allows ions to get through. So a better way to explain that is by looking at a picture here. In the first picture, you can see this is a ligand-gated ion channel. This is just another type of receptor in the plasma membrane. We just saw the G-coupled receptor. Here's the second one. So in this case, the channel is closed until a ligand binds to it. Here's the ligand, and now the channel is open. When it opens, the ions can flow through the channel, and this can send out a chemical response. When the ligand disassociates from the channel, it will close and no ions are able to get through. So this is another type of receptor in the cell membrane. So those were two types of cell membrane receptors, but then we also have to talk about how there are receptors that are actually inside the cell. So what about those little non-polar molecules that are able to diffuse right through the cell membrane? Where do they go? Once they cross, they reach receptors inside the cell, in the cytoplasm, or sometimes even in the nucleus. One common type of messenger that can cross is nitric oxide. Nitric oxide is a little hydrophobic nonpolar gas messenger that's able to diffuse right through the cell membrane. Testosterone is another example. It's a signaling hormone that can cross the cell membrane and bind to the cells, to any cells that have a testosterone receptor inside of them. So they have to have the testosterone receptor in order to receive that message. But the testosterone that gets released from different cells of the endocrine system are going to be secreted. Usually it's cells in the testes for biological males. It's going to travel through the blood, bind to receptors in the nucleus to turn on genes that control male sex characteristics. So here's a better kind of idea of what that looks like with a picture here. Here's your testosterone. So this molecule was secreted by, again, usually cells in the testes that are releasing this signaling molecule. It's going to travel through the bloodstream until it gets to this cell in the picture. This cell is one that is able to receive that message. So because testosterone is a non-polar ligand, it's able to diffuse right through the cell membrane. See in this picture here, there's no G-coupled protein receptor, there's no ion-gated channel, right? It's just going right through the phospholipid bilayer, and it binds to a receptor protein that's in the plasma membrane. So that combination of the testosterone and the protein forms what we call a hormone receptor complex. There's that word again, complex. We see that a lot in biology. That whole system or whole component here enters the nucleus. You can see it went through the nuclear pore. And then it binds to specific genes that are involved in male sex characteristics or male sex genes. It actually kind of acts like a big transcription factor. And this is going to stimulate male sex genes into mRNA, and then they will be translated into proteins that are involved in male sex characteristics. All right, so another type of cell communication, but in this case, this receptor was inside of the cell rather than on the cell membrane. My dog is barking. Sorry. I don't know if you can hear her or not. So after reception, the next stage is transduction. This is a multi-step pathway that involves the activation of proteins by phosphorylation. So you should remember what phosphorylation is, but if you don't, we're going to talk about it. Signal transduction pathways are like the actual falling dominoes. So I kept saying reception as like the push of the first domino. And you can't see me, but I'm actually pushing my hand out because for whatever reason, I think you all can see me, even though I know you can't. So that's like the push of the dominoes, and then all the dominoes falling along that pathway is like the signal transduction pathway. This is going to be a bunch of proteins being activated along a pathway, amplifying a signal. So phosphorylation makes things happen. Do you remember what phosphorylation is? Phosphorylation is when you add a phosphate group to something. In this case, it's going to be proteins along a pathway that activate them or turn them on. The addition of phosphate groups to proteins in the signaling pathway activates them. The phosphates are actually then transferred by molecules called protein kinases. We've seen them before this year. We've seen them in the cell cycle. But they're actually going to be used now to activate messages along a signal transduction pathway. So here's a good overview of what a signal transduction pathway looks like. There's a very similar picture on the signal transduction pathway POGL. Here's the reception part. We've talked about this already, okay? You have an activated, usually an enzyme or protein that's first in the signal transduction pathway. The relay molecule acts as protein number 1 in the kinase cascade or pathway, as they call it. You can see all these proteins along the pathway. The active protein number 1 will then move on and pass its phosphate onto the next and activate then protein number 2. Protein number 2 phosphorylates a protein that brings about the cell's response. In this case, this is a very short phosphorylation cascade. But you get the idea that there would be many proteins along the pathway that are getting phosphorylated or having a phosphate group added to them. And then once they are done, there are another group of proteins called protein phosphatases. I labeled them on the signal transduction pathway or the signal transduction POGL for you, but they will remove the phosphate groups and inactivate them until the next reception or the next signal comes through. So this is all about activating these proteins, again, kind of like the falling dominoes here, falling, adding energy as they fall to the next protein in the pathway, or not actually by falling, but adding phosphate groups or being phosphorylated. So those protein phosphatases that I just described are responsible for dephosphorylating, or basically inactivating, the protein kinases to turn the pathway off when the signal is done being sent. There are, where the signaling molecule that binds to the membrane receptor is said to be the first messenger. So testosterone would be an example of a first messenger. That's something that we just looked at. There are also secondary messengers that are small, non-protein, non-polar messengers that can spread throughout the cell via diffusion. The most common of them are calcium and cyclic AMP or CAMP. You might remember CAMP from our operons when we talked about the lac operon and positive control. So these are secondary messengers that can travel along and inside of the cell to help send messages. We'll look at a specific example. You don't need to know the mechanisms of the secondary messengers, just kind of recognizing that cyclic AMP is one of them, calcium is one of them, and you'll usually see them in pictures where, like this, where they are in the cell being spread throughout and helping to send the message along the signal transduction pathway. Again, you don't need to understand the mechanism here, just that they exist and that you'll see them in pictures as they move along in the signal transduction pathway. So the last stage of this whole process is the response, and this could be any number of cell activities, mainly used in regulating protein synthesis by turning genes off and on, which is why we're talking about this after protein synthesis. So here's a specific example of that. This is a specific example of a signaling pathway that activates a transcription factor that turns a gene on. The response to the growth factor is transcription. So in this case, growth factor sends the message down the signal or activates the proteins in the phosphorylation cascade. This then in turn activates a transcription factor, turns it on, and then that transcription factor is going to bind to the area of the DNA that it's designed to bind to and transcribe the genes. So this would be the response, transcription. This would be like a whole signaling pathway. When we're talking about this reception, transduction, response, we're talking about it kind of in general terms. We're going to look at specific examples in the coming days here of how this actually works. This kind of picture, this idea, this is all over the AP exam. I don't know what it's going to look like this year with only free response questions, but they like to use cell communication in as many possible examples as they can. And it's not that you need to know specific pathways. You don't need to be versed in all the names of all these proteins or anything like that, but you need to be able to kind of recognize and understand this general basic overview of cell communication, reception, transduction, and response. So sometimes a signaling molecule may regulate the activity of a protein rather than its synthesis. So an example could be a signal that may cause the opening or closing of an ion channel in the plasma membrane, something like that. So we're going to look at, again, some examples in the coming days here. And then, of course, there's always the evolutionary tie-in in your book. Evolutionary biologists have discovered that a lot of these cell signaling mechanisms are universal in all organisms, which shows us evidence for evolutionary relatedness of all life. All right, so that is it for section 6. Please let me know if you're having any questions. This stuff is definitely not super easy. So let me know. We're going to do some practice. Keep up the good work.